Hello and welcome dear students. Today we are going to discuss about the topic human evolution, evidences from Y chromosome. The main objectives of today's lecture are to know the basics of human evolution, to discuss about the structure of the Y chromosome, to discuss about the biological functions of the human Y chromosome, to discuss about the role of Y chromosome in human evolution, to discuss about the uses of Y chromosome variation, to discuss about the Y chromosome haplotype evolution, to discuss about the Y chromosome specific microsatellite polymorphisms as well. Dear students, let us first start with the introduction to the topic, that is human evolution. Human evolution is the lengthy process of change by which people originated from ape-like ancestors. Scientific evidence shows that the physical and behavioral traits shared by all people originated from ape-like ancestors and evolved over a period of approximately 6 million years. The evolution of modern humans is a long and difficult process which started from their first appearance and continues to the present day. The study of the genetic origin of populations can help to determine population kinship and to better understand the gradual change of the gene pool in space and time. The past years have seen the increased study of Y chromosome polymorphisms and their relationship to human evolution and variation. Low Y chromosome sequence diversity indicates that the common ancestor of all extinct Y chromosomes lived relatively recently, and the consensus of estimates of time to the most recent common ancestor concur with estimates of the mitochondrial DNA ancestor. Though the reason for low nucleotide diversity on the Y chromosome remains unresolved till date, some of the mutations are proving highly informative in tracing human prehistoric migrations and are generating new hypotheses on human colonizations and migrations. The recent discovery of highly polymorphic microsatellites on the Y chromosome offers new possibilities for the investigation of more recent human evolutionary events, including the identification of male founders. The non-recombining portion of the Y chromosome, that is NRPY, retains a record of the mutational events that have occurred along male lineage throughout evolution. This is because it is inherited patrilineally, that is from the father, as a haploid entity that is not subject to recombination. In this, it is the male counterpart of mitochondrial DNA which records mutational events in female lineage in evolution. Dear students, let us now discuss about the structure of the Y chromosome. The Y chromosome is one of the smallest chromosomes in the human genome, about 60 MB, and represent around 2 to 3% of a haploid genome. Cytogenetic observations based on chromosome banding studies allow different Y regions to be identified. The pseudo-autosomal portion divided into two regions, that is PAR1 and PAR2, and the euchromatic and heterochromatic regions. The pseudo-autosomal regions, that is PAR1 is located at the terminal region, that is towards the end of the short arm, that is YP, and the PAR2 at the tip of the long arm, that is YQ. PAR1 and PAR2 cover approximately 2600 and 320 kb of DNA respectively. The pseudo-autosomal regions, and in particular PAR1, are where the Y chromosome pairs and exchanges genetic material with the pseudo-autosomal region of the X chromosome during male meiosis. Consequently, genes located within the PAR are inherited in the same manner as autosomal genes. The euchromatic region is distal to the par 1 and consists of the short arm paracentromeric region, the centromere and the long arm paracentromeric region. Finally, the heterochromatic region comprises distal YQ corresponding to YQ12. The region is assumed to be genetically inert and polymorphic in length in different male populations, since it is composed mainly of two highly repetitive sequence families like DYZ1 and DYZ2 containing about 5,000 and 2,000 copies of each respectively. Whereas PAR1 and PAR2 represent the 5% of the entire chromosome, the majority of the length of the Y, that is about 95%, is made by the so-called non-recombining Y, that is NRY. This includes the euchromatic and heterochromatic regions of the chromosome. Dear students, let's talk about genes on the Y chromosome and their role in the evolution. Compared to the other human chromosomes, the Y chromosome has a limited number of genes. The Y gene poverty may have been the result of the tendency of Y chromosomes genes to degenerate during evolution, being nowadays the relic of an ancient common ancestry with the X chromosome. Both mammalian X and Y chromosome evolved from ancestral autosomes. The most ancestral gene functions were retained on the nascent X chromosome but deteriorated on NRY portion of the emerging Y, giving females with 2 copies but males with only 1 copy of many genes. The gene dosage problem has been solved through inactivation of one X chromosome in females. In spite of the limited makeup of genes, different transcription units or families of closely related transcription units have been identified in the NRY region during the past decade. All NRY genes can be divided into 2 different categories. The first comprises those genes which are ubiquitously expressed, have X homologs, appear in a single copy on the NRY and exhibit housekeeping cell functions. The second category include genes expressed specifically in the testes, exist in multiple copies with the exception of SRY on the NRY and encode proteins which more specialized functions. Dear students, let's talk about now the biological functions of the human Y chromosome. Several phenotypes have been associated with the non-recombining portion of the Y chromosome. For obvious reasons, most of these are male specific and make the Y chromosome a specialized chromosome during human evolution. The most characterizing feature of this chromosome remain its implication in human sex determination and in male germ cell development and maintenance. Dear students, let's talk about the SRY gene and sex determination. The first indices that the Y chromosome was involved in male sex determination came from the observation that XY or XYY, that is Klinefelter syndrome, individuals develop testes, where XX or X0, that is the Turner syndrome, individuals develop ovaries. In 1990s, the genes responsible for testicular determination, named SRY or sex determining region on the Y chromosome, was finally identified. SRY was cloned by isolation of small fragments of translocated Y on XX sex reversed patients. This gene is located on the short arm of the Y chromosome close to the pseudoautosomal boundary. It comprises a single exon encoding a protein of 204 amino acids which presents conserved DNA binding domain, the HMG box, that is high mobility group. suggesting this protein regulates gene expression. This gene has been shown to be essential for initiating testes development and the differentiation of the indifferent bipotential gonad into the testicular pathway. Moreover, SRY has been proposed to be the master gene regulating the cascade of testes determination. Dear students, let's talk about now oncogenic role of the Y chromosome. The implication of the Y chromosome in cancer remains still speculative. Y chromosome loss and rearrangements have been associated with different types of cancer such as bladder cancer, lung cancer, and esophageal carcinomas. Although loss and rearrangement of this chromosome are relatively frequent in different types of cancer, there is no direct evidence for a role of Y chromosome in tumor progression, since no proto-oncogenes, tumor suppressor genes, or mismatch repair genes have been localized to the Y chromosome. Dear students, let's now discuss the evidence of evolution. Darwin and the 19th century biologists found compelling evidence for biological evolution in the comparative study of living organisms in their geographic distribution and in the fossil remains of extinct organisms. Since Darwin's time, the evidence from these sources has become considerably stronger and more comprehensive, while biological disciplines that emerged more recently, like biochemistry, genetics, physiology, ecology, ethology, and especially molecular biology, have supplied powerful additional evidence and detailed confirmations. The amount of information about evolutionary history stored in DNA and proteins of living things is virtually unlimited. Scientists can reconstruct any detail of the evolutionary history of life by investing sufficient time and laboratory resources. Evolutionists no longer are concerned with obtaining evidence to support the fact of evolution, but rather are concerned with what sort of knowledge can be obtained from different sources of evidence. Thousands of human fossils enable researchers and students to study the changes that occurred in brain and body size, locomotion, diet, and other aspects regarding the way of life of early human species over the past 6 million years. Study of human genetics show how closely related we are to other primates, in fact, how connected we are with all other organisms, and can indicate the prehistoric migrations of our species. Homo sapiens all over the world. Exciting scientific discoveries continually add to the broader and deeper public knowledge of human evolution. Dear students, let's now talk about the role of Y chromosome in human evolution. Apart from the uses of Y chromosome analysis in forensics, the Y chromosome has given a major contribution to the understanding of population histories. The Out of Africa model proposes that all modern humans can be traced back to a single ancestor that lived in Africa and that dispersed throughout the other continents, replacing archaic human species. This contentious issue in human evolution, first indicated by mitochondrial DNA data and later supported by other mitochondrial DNA and autosomal studies, is generally corroborated by global Y chromosome phylogenies showing greater diversity in African populations than in other, and placing the first branch between exclusively African populations and African and non-African populations in the phylogenetic trees. Using a combination of rapidly evolving microsatellites and or more stable binary markers, substantial continental and local population structure has been revealed, and hypotheses of early human migrations confirmed or formulated out of African and back again. Novel insights into other major human dispersals have been gained, such as the colonization of the America, different migration events in Asia, as well as the demographic history and genetic structure of different populations over time. However, the Y chromosome records only the paternal history of a population, and in comparison to mitochondrial DNA, it can provide information on related but different aspects of the same genetic history. Discordance between mitochondrial DNA and Y chromosome variation occurs and may be due to factors which influence the spread of males and consequently of Y chromosomes in human societies. African populations show the largest amount of genetic variation. Genetic diversity outside Africa is derived from a small number of African ancestors. Although this view is generally supported by Y chromosome phylogeny as well as by mitochondrial DNA, and autosomal data discrepancies occur in the estimations of the time of the most recent common ancestor between the different systems. Application of the coalescence approach, which assumes that natural selection is not affecting DNA sequence variation, yields values of, for example, 535,000 years for a non-coding region at XQ and 800,000 years for genes encoding beta globulin and 1,860,000 years of the pyruvate dehydrogenase or PDHA1. In the case of the Y chromosome, although initial Y chromosome studies gave a coalescence estimation similar to mitochondrial DNA studies, a recent study using many biallelic markers in a large worldwide chromosome set estimated a time of 59,000 years. This value is in close agreement to estimates based on the Y microsatellite data. A number of factors can explain these discrepancies, which include: 1. That different number of chromosomes and markers analyzed in the different studies. 2. Effective population sizes of the X, Y chromosome, mitochondrial DNA and autosomes, that is the effective population size of the Y chromosome is 1/4 that of the autosomes, and therefore autosomal lineages are expected to take 4 times as long to become extinct. 3. That different methods and assumptions used in estimations of the age of the MRCA, that is exponential population growth model, versus constant population model. And 4th, that is different selective pressures acting on different loci. Even taking into consideration these factors, the Y estimate is still more recent. In loci that do not experience recombination, such as mitochondrial DNA and Y chromosome, natural selection, either positive or negative, acting on one single locus will consequently act on all others, altering therefore the history of the whole molecule. However, it must be pointed out that calculations of Y chromosome coalescence times are far from clear-cut. Further studies reanalyzing all comparable and available data, taking into account the same demographic and theoretical assumptions, are urgently needed to reveal the extent to which selection may have contributed in shaping the patterns of variability at different loci, that is the Y chromosome, mitochondrial DNA, autosomal loci, during the course of evolution. Dear students, let's now discuss about the uses of Y chromosome variation. The NRY and the maternally inherited mitochondrial DNA do not undergo recombination, and therefore they have inherent advantages, that is straightforward construction of individual haplotypes, single paternal maternal ancestry, easy estimation of the timing of branching events in phylogenetic trees using calibrated mutation rates, etc. That makes the interpretation of human genetic history is less complex than that deduced from the analysis of autosomal genes. An increasing number of informative Y polymorphic markers are available for population and forensic studies, and including base pair substitutions, insertion deletion events, microsatellite and mini satellites. Both biallelic variants and insertion deletion polymorphisms almost certainly represent unique molecular events in human evolution, and although in some cases that can be signatures of small groups or individuals, can define deep rooted phylogenetically related haplogroups with either a wide geographic distribution or in some cases more regionally clustered. Microsatellites or short tandem repeats STRs define the variation, that is haplotypes, within haplogroups and allow separations of human populations to be more accurately measured, as well as the coalescence times of well defined haplogroups to be estimated. Dear students, let us now discuss the Y chromosome haplotype evolution. Research groups have used the few unique mutational events to construct haplotypes for both tracing the evolution of Y chromosome and detecting any geographical pattern in their distribution. Scoring 9 such events from worldwide populations has shown that most Y haplotypes are population specific and that each population exhibits a characteristic suite of Y haplotypes. Parsimony analysis generates a single tree of Y haplotypes which exhibits a star-like pattern which is similar to those often seen with empty DNA haplotypes. Dear students, let's now discuss the Y chromosome specific microsatellite polymorphisms. Several studies have illustrated the value of Y specific microsatellites or short tandem repeat polymorphisms in studies of human evolution and variation. Firstly, whereas there is a dearth or shortage of sequence and even mini satellite polymorphism on the Y chromosome, this is not the case with microsatellite, which shows levels of diversity similar to their autosomal counterparts. Because of recurrent mutation, these markers do not display disequilibrium as strongly as sequence polymorphism, and the pattern of allelic variation appears to result from the same evolutionary events proposed for their autosomal equivalents. The most studied Y-specific microsatellite, that is DYS19, a tetranucleotide repeat, exhibits considerable heterogeneity both on a continental and regional scale. Variation among Europeans, even between neighboring countries, can be highly significant, but there is, however, some conflict as to whether there is detectable structure to this variation. A study has identified a decline for some DYSZ9 alleles in Europeans, whereas another study has found an absence of pattern in allele distribution in their samples of European populations, leading the authors to suggest that this randomness is caused by tetranucleotide repeats evolving faster than dinucleotide repeats, which did detect structure. The power of using Y microsatellites in the form of haplotypes, however, to discriminate between closely related groups has been demonstrated. Attempts to infer evolutionary relatedness among these haplotypes by using the principle of maximum parsimony have been unsuccessful because of the extremely high number of tree permutations. Applying a model that is based on molecular variants developed for analysis of mitochondrial DNA sequence data demonstrated a significantly larger mean proportion of between population than within population variance. This finding of structure using trinucleotide and tetranucleotide microsatellites is in contrast to a study of Y-specific dinucleotides in small samples of world populations, which indicated that any tree of Y chromosomes is shallow because the distribution of alleles are similar among the populations. Together with mitochondrial DNA, studies of Y chromosome microsatellite loci will help identify the number of male founders of particular historical isolates. Dear students, let's now conclude the lecture. Evolution is the process by which nature selects from the genetic diversity of a population those traits that would make an individual more likely to survive and reproduce in a continuously changing environment. Evolution stresses the relatedness of all life rather than its differences. The human Y chromosome is strictly paternally inherited and in most of its length does not recombine during male meiosis. These features make the Y chromosome a very useful genetic marker for different purposes. The Y chromosome is an important component of the human genome with an essential biological function such as mammalian sex determination and male fertility, and in studies of the origin and genetic relationships between human populations. Dear students, in the last decade, the Y chromosome has been increasingly used to investigate the evolution, migrations, and range expansions of modern humans. The possibility to construct highly informative Y chromosome haplotypes has also had a significant impact in forensic studies and paternity testing. However, recent experimental and statistical analysis suggests that both positive and negative selection are acting on the Y chromosome and consequently may influence Y chromosome haplotype distribution in the general population. Dear students, it was all about today's lecture regarding human evolution, evidences from the Y chromosome. Hope you have understood it well. See you next time with a different topic. Till then, take care and bye bye.